Okay. Good morning, everyone. Thanks for joining us this morning for Grand Rounds. My name is Jonathan Duska. I'm one of the neurology chief residents, and it's my great pleasure to introduce this morning's speaker, Dr. Cenk Ayata, who's a professor of neurology, a member of our stroke service, and director of the Neurovascular Research Unit here at MGH. Dr. Ayata is a vascular neurologist and a clinician scientist engaged in translational neurovascular research in brain injury, cerebral small vessel disease, and migraine for over 25 years. Dr. Ayata received his MD degree from Hacettepe University in Turkey, following a postdoctoral fellowship at the Massachusetts General Hospital. He completed his residency training in neurology at Tufts University and a clinical fellowship in vascular neurology and neurocritical care here at MGH. His laboratory uses electrophysiological and multimodal optical imaging techniques in animal models of ischemic or hemorrhagic brain injury and migraine to dissect pathophysiology and test therapeutic interventions. His work has been supported by the NIH, AHA, Leduc Foundation, industry, private nonprofit foundations, and philanthropy. In nearly 200 peer-reviewed publications, his most notable contributions have been on spreading depolarizations in ischemic stroke and migraine and preclinical testing of stroke therapeutics. So without further ado, I'll turn the floor over to Dr. Ayata, who will be speaking about spreading depolarizations in clinical neurology. Time to call it what it is.
All right. Thank you so much, Jonathan. I've been here as an attending for almost 20 years now, and I've been invited to give the grand round several times. It never worked out because of scheduling conflict. So this time the stars were aligned, and I'm here giving the talk. Finally, better late than never. And I picked a topic today that is. All right, how do I change this slide? All right, there we go. Okay, that is both dear to my heart because of the research I do, and also something that comes up quite often during our daily rounds on inpatient services. The term spreading depression, probably most of you have heard about, but the residents, fellows, and even attendings, when I get down to it, you don't really know what it is and what to do with it in the clinical setting when you come across a patient with telltale signs of spread depression. A typical scenario is that the resident presents the case with symptoms suggestive of spread depression, and at the end I say, okay, what do you think it is? You know, what is your analysis? And there's a moment of awkward silence and some hesitation, and the response is usually, is it that spreading depression thing? And I say, yes. So the main purpose of my talk today is to try to help you shed this anxiety of talking about spreading depression and to give you some more confidence so that you can start using the term spreading depression in the clinical setting. Okay. All right. Now, to that end, what I'm going to do is give you a simplified overview of spreading depression physiology at the cellular level and membrane level, ionic level. And then I'm going to list a number of clinical conditions associated with ST that we know of. Then I'm going to move on to ST semiology so that you have an idea what to look for when you're thinking about spreading depression. And at the end, I'm going to make a case. It won't be an easy sell, I know, but I'm going to make a case to introduce the term spreading depression in routine clinical practice, which is not the case at the moment. So, okay, let me start with this now really iconic figure from Leal's paper published almost 80 years ago now. He was a visiting scientist at Harvard Medical School, not too far from here, when he was working on epilepsy mechanisms. He was using rabbits for his experiments, and when he placed a number of electrodes serially across the cortex, as you see right there, and when he delivered a really strong depolarizing electrical stimulus, where you see S right in the frontal pole, instead of getting the intended seizures, he ended up more often than not getting this profound depression of EEG. And what was most peculiar was that this depression then really slowly propagated from the stimulus site across the cortex at a really creeping, crawling speed of 3 millimeters in a whole minute. It's this much, 3 millimeters in a whole minute. When we think of electrical activity, this is super slow, right? And propagated from one electrode pair to the next, all the way to the back of the cortex. And then it recovered in the order that it was depressed within a matter of about 5 to 10 minutes to come back to normal. Now, naturally, he called this spreading depression. That's where the name is coming from, okay? I'm pretty sure he was super annoyed by this, as I was 30 years ago when I was trying to do hippocampal electrophysiology and seizure mechanisms. Because when you get this, the prep is lost. You have to start from scratch all over and discard that prep, because even though the cells may still be viable, they don't behave the same way. So you have to sort of scrap it out. But he was a smart person, as you would expect. He said, if I can't prevent this, I might as well study this for the rest of my career. And that's what he did. He published a series of really influential papers and essentially opened up a new field with these papers. He remains the most highly cited paper, the author in spread depression field to date. Now, since his times, we actually had a lot more to do, of course, and we learned a lot more. So we now have a basic understanding of what is going on during spread depression. And I know we have electrophysiologists here, and I'm underscoring this. This is a very simplified model. It's not as simple as this, but to help everybody understand this, I have to simplify it. So these are a whole bunch of neurons, and I'm not an artist, so just excuse my skills. As you all know, the ions are unevenly distributed across cell membranes, right? Intracellular potassium is much higher than extracellular potassium. Calcium and sodium are also unevenly distributed, but in the opposite direction. Extracellular is higher than intracellular levels. And when you deliver a really strong depolarizing stimulus at a focal point in the brain, which then elevates the extracellular potassium levels to more than 12 millimolar in about 1 millimeter cube of brain tissue, that's gray matter. 1 millimeter cube, 12 millimolar threshold for extracellular potassium. This suddenly triggers a transformation of neuronal cell membranes. They become very leaky because of opening of a large non-selective cation conductance that then allows all these ions to flow across their or along their concentration gradients across the membrane. As a result, potassium flows from inside to outside the cell. There's a massive potassium efflux, elevating the extracellular potassium to about 20 to 60 millimolar levels. Now, those of you who do electrophysiology know that extracellular potassium is extremely tightly controlled in the brain. 3 to 4 millimolar is the resting state. Even the most intense seizure may not get elevated beyond 8 to 10 millimolar level. Yet during spread depression, potassium goes up to about 20 to 60 millimolar levels. At the same time, there's a massive calcium influx. Together with that astrocytic reuptake pump reversal, there's a massive glutamate elevation in the extracellular space. Okay, so it's not just the uncontrolled synaptic release, but reversal of the pumps dumping a glutamate from astrocytes into the extracellular space. So now we have a massive glutamate increase in the extracellular space. At the same time, sodium flows into the cell, pulls in chloride and water passively, we believe, and the cells swell. This cell swelling is very similar. In fact, we cannot distinguish the two to what happens during ischemia. The same dendritic beating and swelling happens during spread depression. The difference is it's perfectly reversible without any residual injury to the cell. Okay. Now, what we believe happens next is that potassium and glutamate, the two most potent depolarizing agents in the brain, slowly diffuse from the point of release through neighboring cells. Okay. Now the neighboring cells are bathing in 20 to 60 millimolar potassium and elevated levels of glutamate, which then triggers the same depolarization cycle, membrane conductance in those cells. Now they dump their own potassium and glutamate. And that diffuses to the next layer of cells. And by this way, by way of chemical contiguity, spreading depression spreads. Okay? So that's why it's called spreading depression or spreading depolarization. In essence, it is a depolarization, not really suppression of the cells. Because the cells are massively depolarized to 0 millivolt, essentially, you cannot have action potentials, you cannot have synaptic transmission, so you cannot have electrical activity the way we know it, right? EEG is flat. Now, what is really equally puzzling to us, even today, is that within less than a minute, cell swelling, all the transmembrane ion balances are restored by way of ATP-dependent pumps, reuptake mechanisms, and both on astrocytes and neurons, as well as vascular clearance of extracellular potassium out of the tissue. Everything comes back to normal. And that's why it's really completely reversible. It's not injurious to the brain by itself. So, they say a picture is 1,000 words and a movie is 1,000 pictures. So you get the idea. What I have here is a movie that hopefully will show. I don't know if it will show. Earlier, it created some problems. Let me get out of this. Okay. Oh yeah, there you go. You come on, don't do this to me. We tested it just before you guys walked in. I can't believe this. Not giving up. All right, well, I have another movie. If this doesn't work, I have another movie. Say it again? Yeah, I'm trying here. Yeah, it doesn't work. Okay, well, anyway, so I have a very similar movie down to 2 slides. I did that. All right, let's go. Let's not waste time. Let's keep going. Okay. Trust me. Okay, I have another movie. That's okay. So hold that spot in your head. I'm going to have another movie later. Okay. Okay. All right. Now, all these ion shifts create a characteristic extracellular electrophysiological signature for spreading depression. Okay. Now, remember, any kind of intense depolarizing event that raises the potassium to more than 12 millimolar in the brain tissue is capable of triggering a spreading depression. That could be chemical, like applying topical potassium chloride from outside, like in the experimental setting. It could be electrical, like Leao did. Or it could be mechanical distortion of the brain tissue. If you take a pinpoint and just gently touch, not injure, gently touch and indent the cortex, that is capable of triggering a spreading depression. Ischemia is equally depolarizing, so that can trigger a spreading depression. And seizures, we know, can culminate in spreading depression because of the potassium threshold. Regardless of the trigger, when there is a spreading depression triggered, it propagates across the cortex in the mouse or rat cortex, which is lissencephalic, including the rabbit cortex, in virtually every direction from the point of triggering. Okay. And typically it reaches, these are all messed up, but they are supposed to be serially placed electrodes like that. It reaches the first electrode, which is what you see here. Okay. The ECOG depression is the same as Leao observed in his rudimentary amplifier setup. But what is more important is that there is a massive extracellular depolarization signature that is a very large, and this is the amplitude scale here you should pay attention to. This is about 20 plus millivolt slow negative potential shift picked up by an extracellular glass micropipette. Okay, and lasts about a minute or so, typically less than a minute, as I showed you earlier, right? And this is the electrophysiological signature of spreading depression that we always look for in the experimental setting and in the clinical setting when there is an intracranial electrode. Now, After hitting the first electrode right there, it propagates at this speed to the second electrode, which then gives you the same electrophysiological signature with a latency, as you can see here. Now, this is a self-regenerating process. This is not I shout and then my voice travels and dissipates over time. It regenerates itself. And in fact, if you just look at the amplitude of the second wave farther away, it's not any smaller. In fact, it's a little bit bigger than the first one. It did regenerate itself as far as it could go until it hits a barrier. This barrier typically is a white matter trap, where there's a lot of extracellular space and not too many neurons to release their potassium and to get depolarized, and there's not much glutamate in the white matter either. So there is no fuel to regenerate the wave, and it dies off. You can think of this as a wave hitting the beach, right? Beach is the white matter, and just splash it into, and then it just dies off, doesn't regenerate itself. I resemble this to dropping a pebble in a pond. It sort of helps people think about this. The pebble would be the depolarizing stimulus, and the waves go out. Now, if this doesn't work, I'll be really pissed. Okay, I'm officially pissed. All right, I don't know what's going on here. Guys, I'm sorry. I don't know what's wrong with this, but these are supposed to be neuronal intracellular calcium imaging using 2-photon microscopy to show you a wave washing across the cortex and to give you an impression of what the spread depression looks like as it spreads. You can find these movies in the publications below, and I'll just move on, unfortunately. So all these changes, and I told you that it's a massive potassium release, massive glutamate release, but it's not limited to that. Every neurotransmitter or neuromodulator that has been measured to date also goes up, because everything is bathing in or washed in extracellular potassium levels of 60 millimolar, which then essentially depolarizes every single cell. This is not only neurons and astrocytes. We're talking about microglia getting depolarized. They're not excitable, but their membranes get depolarized. Perivascular nerves will get depolarized and release their own neurotransmitters that they're releasing onto the blood vessels. Smooth muscle cells, endothelial cells, pericytes, everything is bathing in the same level of extracellular potassium. So because of that, it really wreaks havoc in the tissue, but not injure the tissue. Gene expression changes, morphology changes, blood flow changes, metabolism changes, everything changes, but reversibly so, okay? And that's why it has been called a brain tsunami. The top thing is blocking that title there. Yeah, okay, well, this is supposed to be a movie of the topical potassium chloride application-induced brain depression. I mean, is there a way to fix these movies? I'm sorry, I mean, I have like 10 different movies to show, and the whole idea is
yeah, I don't know.
and we tested it outside, right? I don't know why it's not showing. Maybe change from the VGI or VGA, whatever it is, to HDMI again.
Oh, well, we could try, but we weren't getting any connectivity. I don't think that's it. I think the problem resides with your files working on this piece.
You're right,
You tested it outside.
because we changed to the VGA. The HDMI didn't work.
You tested it and it worked. Did you restart?
Okay, well, we can restart the PowerPoint. It takes very little time. You want to do that?
Okay, yeah, I guess we could.
Let's escape that.
And here we're gonna close.
All right, well. So let's begin.
The second one, doing that, is there a change in the volume of the accelerator debate?
Yes, extracellular space shrinks dramatically. And that we believe contributes to the elevation in extracellular concentrations of potassium chloride and glutamate. So that's an important point. In fact, in tissue where extracellular space is large in proportion, like white matter, the strength threshold becomes much higher. So it's harder to induce.
Yeah.
So
It
HDMI.
often terminates itself.
No, it is the issues. No, it's
In fact, if you apply hypotonic solutions extracellular to make the cells swell,
All right.
that
Let's,
by itself can trigger spread depression without any other depolarizing agent. Even though cells are not necessarily depolarized, the shrinkage in extracellular space does it trigger, and there is a spread depression. It triggers from that. And that's a common experimental problem that the beginners have.
This may be for the end of the talk.
Yes.
Do you think there's an evolutionary value of this?
Jesus, that's a huge question. Yes, yes, absolutely. So we actually have published a couple of papers so far. We are actively looking into that. The question is, such an intense phenomenon, right? Why is it still there? If you look at any mammal, move on down, reptiles, move on up, maybe a little bit birds, and then even locust central nervous system has spreading depression imprinted in it as a mechanism.
So the other possibility is just that there's some physical data causes, it didn't evolve.
Well, yeah, so there are two ways of thinking about this. You know, it's a disease process, right? We consider it as a disease. The alternative is that it serves such an important purpose that if it was eliminated by evolution, it would be worse, right? And one example for that we recently published, I think a couple of weeks ago now, is that depression actually in vivo works to terminate the seizures. What the seizures trigger spread depression. And if you actually trigger spread depression from outside, it terminates the seizure. And the anti-epileptic effect is not just a short period of that depolarization. That would be expected, right? It lasts more than 30 minutes. Long after depolarization is gone, cells are back to their normal membrane. And that's one of the working hypotheses that we have been working with. The other setting is that spreading depression constricts blood vessels in the brain. It causes mild spasm like that. And one of the settings that spreading depression is triggered is trauma or intracerebral hemorrhage, right? Now, we believe that spreading depression helps control hemorrhaging when there's either traumatic or primary CH. We did an experiment on the papers under review now, stroke. When you induce spread depression from outside in an ICH mode, in a collagen-induced ICH mode, for example, hemorrhage is limited to 50% of the expected size, which you induce spread depression from outside. So, and when spread depression is triggered by the ICH itself, the growth of the hematoma stops like this, plateaus. So there's a lot more data coming, hopefully, in that regard, where evolutionary selection advantage for spread depression is explored. There has to be a reason for it. Yeah.
In the last few years, last maybe 3 years, people starting to equate the post-ictal depression of consciousness to spreading depression. I mean, you see like you can see a little bit of shift.
Yeah. So it could be, but remember, spread depression is a highly potent process. You don't get spread depression removed with every. So if it is triggered in a case where you might expect the level of consciousness to be affected, yes, by all means, that might be the reason. But you have to think about other things. After a seizure is terminated, there's open a lot more adenosine and whatnot. The synaptic network function is also messed up. So maybe that has something to do with it as well. So I can't blame spread depression just by itself.
But I didn't understand, like you said.
So
I didn't understand, you said that the thing that stops it from spreading is regenerative. The thing that can stop it from running into a white matter.
yes,
If you think about the cortex as a sheet of
yes.
gray matter, has that spread?
In animals it does. Do we have any hope for this? Because I feel like we're at the end of the talk. I have never had such a long time at the end of any talk to answer the questions. That's great. It should be all debate discussion. So it does. Now, the susceptibility to spreading depression is not an absolute thing. There's great gradation here.
No,
As you go higher in the evolutionary tree, as the brains get more and more complicated, for example, in mouse brain versus the human brain, the mouse brain is much more susceptible to spine pressure. The thresholds are much lower to trigger it than a human brain. And we contemplate the reason maybe the astrocyte neuron ratio, because astrocytes typically clear out buffer the extracellular potassium, hit the brake on the excitation and whatnot, right? So that's what we are thinking, that may be the cause. In human brain, therefore, when it's treated, it doesn't necessarily go all the way through the sulfide. Okay, we're back. Thank you so much. Yeah. Oh, I lost all. And if I'm late at the end, don't blame me. Okay, all right, excellent. So you're just seeing this expression in maybe can we cut down the lights? Turn on the lights a little bit. Oh, very. What is the slide? Okay. So anyway, I talked about this slide. Here's a rerun of the same thing. This is the intracellular calcium, and it's just a wave that washes across the cortex.
What's the time?
Yeah, this time span is set up, and this is all of it. It's small field, probably about 500 micron by 500 microns, so it's hard to judge the propagation rate. But the speed of propagation is 3 millimeters in a minute. So if you do that 3,000 microns in 60 seconds, 50 microns per second, so 1 neuron per second kind of jump. You just keep that in that scale. And that propagation speed is also not constant. In hippocampus, for example, propagation speed is much faster because of the cycle architecture of the cell. It's much densely located and reaching in receptors and whatnot. Okay, can I?
Yeah, go ahead.
So am I running this or this?
Yeah, you're running this. Keep running this.
Keep running this. Okay. All right. So that's why it has been called the brain tsunami. All right. Here is a movie from a mouse cortex. We are now scaling up. The movies that I showed you earlier are 2 photon movies where you're looking at essentially at a sort of a cellular level or a small field level. This is an entire mouse cortex. Let's run it one more time. The imaging field is located over the right hemisphere to cover the entire hemisphere, and spread depression is triggered with topical potassium chloride on the upper right corner of the field right there, okay? And this is how it spreads. The movie is again sped up. You can see the time on the top of the movie, okay? And this is blood flow changes. It causes significant constriction as it propagates. And this is, now we are scaling even more up. We're now looking at a gyroencephalic brain, very similar to human brain, but it's a pig brain, okay? The field is located right in the middle of the dorsal head. The midline is right there. This is the midline, okay? This is a dura folded, reflected. And these are sulci and gyri and the blood vessels and whatnot. The spread depression is going to be triggered by topical potassium chloride application in this movie, okay? And here's the application. Here's how the spread depression propagates in the pig brain. What you will sort of see is that it prefers to ride the gyri as opposed to dip into the sulci, okay? It does dip into the sulci every now and then, but there's a tendency to stay on the gyri. But I want to imprint this image in your head, right? When you think about a patient, when you see a patient describing symptoms propagating from one part of the body to another, or a visual aura, the scintillating scotoma, imagine this wave moving over the gyrus and sometimes jumping the sulcus. Say it again.
Is it regarded or just?
So this was the induction, and this is one wave. And there may be re-entrant waves coming backwards. One induction can lead to re-entrant waves. Is that what you're asking? Recurrent waves. I'm having a hard time hearing you. Can you speak up? Just come down. Yes. Just come to the front. There's plenty of room here. All right, fine. That's fine. Okay. So now, massive ion changes, electrophysiological changes, massive depolarization, right? Massive increase or decrease in blood flow, metabolic changes. Why don't we see it in the clinic more often? Why has it been so elusive for us, right? Spread depression. We see seizures all the time, right? We see them on the EEG, we see them sometimes on the imaging. Why can we not see this? Because there are a lot of barriers to our detection of it in the clinical setting. Let me explain this with this cartoon. Let's say, this is hypothetical, right? Let's say there's a spread depression triggered on the occipital pole right there, and it's propagating anteriorly this way. Each band that you see here, colored band, represents some form of perturbation that the ST induces in the tissue, okay? Let's focus on the first one, the yellow one that says DC shift, right? Now, if this DC shift is 25, 30 millivolt in amplitude, why can't we detect it on EEG, scalp EEG? 20 millivolt, we should be able to see something. Well, it only lasts 1 minute, and it propagates at 3 millimeters per minute. So at any given time, only 3 millimeters of brain tissue is depolarized. That 20, 30 millimolt signature is coming from 3 millimeter strip of brain tissue. And then there are gyri and sulci. In other words, it's not a flat surface to amplify the dipole, right? So we are unable to see this using conventional scalp EEG because of spatial summation, okay? The second reason is the DC shift is very slow, as I showed you earlier, right? It's a very slow wave, builds in about 3 to 5 seconds, stays there steady, and then recovers in about 5 to 10 seconds, which is too slow for our conventional EEG amplifiers, which use high-pass filtering. So it's completely filtered out from any EEG that you're looking at, unless it is a DC amplifier that you're using, okay? So for that reason, DC shift is not detectable. The same thing applies in terms of spatial summation to EEG depression. Now, look at this. EEG depression is about 5 minutes lasting, right? So at any given time, 3 millimeter per minute spread, 15 millimeters of brain tissue is undergoing EEG depression. But the pickup area of a typical scalp EEG lead is about 8 to 10 centimeters square, right? So it's still spatially summated, and there's a lot more activity coming in. So typically, this is below the threshold of detection using conventional EEG. I told you that the cell swelling during spread depression leads to, is similar to what you see in ischemia, right? So it actually causes diffusion restriction in the brain tissue. Why can't we use that? Well, we can't because that only lasts about 2 minutes, and 3 millimeter per minute spread rate, it's about 6 millimeters strip of brain tissue that is depolarized or diffusion restricted at any given time. 6 millimeters. That is really below the detection level of our conventional MRIs. You have to have a high field strength MRI. The second problem there is that the wave is not stationary. So you can't keep imaging and increase your resolution by averaging because the wave is moving. There is not only spatial averaging, volume averaging, but also temporal averaging going on. Okay, so, and the other changes are not usually typically monitored in the clinical setting, so they don't really help much. And for these reasons, today, detection, absolute, incontrovertible detection of ST depends on intracranial recordings using DC amplifiers. Or you have to try a high field MRI and really hope that you get lucky. Short of either one of these present, you have to rely on ST semiology in the clinic to be able to detect it. Now, despite these barriers, today we have a long list of diseases that we have evidence, direct, incontrovertible evidence for ST occurring in human brain. Okay, I have two tables here. The one on the left is the list of diseases we have definite evidence, either electrophysiological or imaging evidence of spread depression in human brain. And the one on the right is in the absence of electrophysiological or imaging evidence, but semiology reported in detail and congruent with what you would expect ST to cause. Okay, and multiple corroborating reports in the literature. I'm going to go over these in more detail, so you don't have to worry about individual things. I will start with the left table, definite table, and I will start with migraine aura because it is the prototypical ST disorder, essentially, okay? So the typical visual aura, the migraine aura is a visual aura, which we call scintillating scotoma. Typically starts at the focal point, usually near fovea, spreads out from that. And scintillating scotoma term comes from the fact that at the wavefront, there is just a fortification pattern, scintillating or shimmering quality to it, in the wake of which you end up getting loss of vision, which is why we call it a scintillating scotoma, okay? And as early as 1941, Lashley drew his own aura, and he was astute enough to know the cortical retinotopic organization, and he calculated his own symptoms, propagation rate as 3 millimeters per minute in the occipital cortex. 1941, even before spreading depression was actually described. Okay, and since then we have accumulated a lot more evidence suggesting that migraine aura is indeed caused by spread depression. Okay, but what really nailed it down is this study by Nushin Hajikani and my longtime mentor Mike Moskowitz here at MGH. When they captured a migraine aura, visual migraine aura in fMRI, okay, the patient had a predictable trigger, and they put the patient in after the trigger was delivered, and the patient had this perception during the aura event. And what you see here down is the retinotopic mapping of this patient, okay, based on the visual field mapping onto the cortex. Putting the two together, they were able to detect a slowly propagating bold signal disturbance across the occipital cortex, visual cortex in this patient. And this is really a direct imaging demonstration, and maybe the only one of a spread depression in action. There are only a few skeptical holdouts left out in the world, I think, who don't believe that spread depression is the underlying cause, but pretty much everybody else believes that migraine aura is spread depression. So let's move on down the list, on the definite list, right? Migraine aura is spread depression. But if you just pay attention to everything else below migraine aura, they are pretty much diseases where you have a clinical indication for a craniotomy to justify putting in electrodes directly over the cortex, subdural electrodes directly over the cortex, or intraparenchymal needle electrodes, okay? So let's take a look at them really quickly. Here's an example from ischemic stroke, malignant MCA infarct, as you see down here, hemicraniectomy to save patient's life. A lead is placed, a strip electrode is placed, as you see there, on the frontal end or anterior end of the lesion, and spread depressions are detected, as you can see here. Okay, now these are still not as crisp as what you would see with a glass micropipette, because there's still, the electrode is about 5 millimeters in diameter. There's still some spatial averaging going on, and the amplitude and the temporal structure is lost there. But these are spread depressions moving one lead to the next, accompanied by EEG depression. Okay, here's a cluster of spreading depressions developing in this patient between 48 and 58 hours after stroke onset. Okay, and there are two clusters, one moving in that direction from electrode D to A, and one moving from A to D in direction. Okay, you can easily map these out. I'm going to show you another pig movie. Hopefully this is going to work. Yes. Okay. So here what we have, instead of potassium chloride, here what we have is a focal cerebral ischemic lesion by MCA occlusion. The black outline shows the ischemic territory. What you just saw at the beginning of the movie was a spread depression wave, and this is the second one in this pig. Now remember, these movies are really sped up by 10 plus times. So the waves that you're seeing don't propagate that fast. They are really very slowly propagating waves. But they do originate from the ischemic lesion spontaneously. Here, there's no exogenous trigger but the ischemia itself. Here's an example from aneurysmal subarachnoid hemorrhage. Intracellular strip electrodes and detecting the spread depolarization wave going from one electrode, one side of the strip towards the other side sequentially, okay? And the DC shift, slow potential shift is flipped here because of the clinical convention of negative being up. Otherwise, it's a negative potential shift. Here's an example from intracerebral hemorrhage. Massive lobar hemorrhage, a strip is placed right around here, and you can see the depolarizations. These are small, slow potential shifts or small, slow potential changes, SPC they call it, moving from one electrode to the next, sequentially associated with EEG depressions. Let's not forget TBI, massive TBI, contusion, hemicraniectomy to save the life again. 2 cases are shown here, 2 strips placed different in each patient, and each one detects the spreading depolarization waves. This is just to give you an idea of the extent of the evidence. They all look the same. They are spreading depression waves. And more recently, we have evidence that acute or chronic subdurals develop spreading depressions. Okay, and we see that quite often actually on our consult service when patient develops recurrent migraine aura-like symptoms, and we freak out. You know, is it a seizure? Is it this? Is there ischemia going on? Reaccumulation of the subdural? What is it? We don't know, but they are triggered. This is a patient with chronic subdural evac, and a strip is placed as you see there, and the depolarization wave is recorded.
After the hemorrhage is dependent.
This was within the next 48 hours. But in subdural cases, we don't have enough evidence. I can only point to maybe a couple of cases directly demonstrated.
So the waves keep on going on.
Right. So yeah, the waves keep on going on. But so let me just give you a little bit different statistics. Ischemic stroke and aneurysmal subarachnoid hemorrhage develops ST pretty much 100% of all the patients, okay, as a disease. In subdural and TBI, we believe, and intracerebral hemorrhage, we believe it's 50% or less. Not everybody develops ST, okay? When they develop, the ST depends on the underlying condition. Again, in subdural, I don't have enough evidence to just give you an idea how long they continue in case of subdural. You know, when do they start? When do they stop? I can't tell this, but in intracerebral hemorrhage, typically this is within the first 48 hours and extending into, I think, 5 days, but I can get back to you on that one after looking at the paper. Okay, well, that's a good question. How long does it continue, right? In subarachnoid hemorrhage, it continues up to 7 to 14 days. In fact, that's why we believe there is a connection with delayed cerebral ischemia or vasospasm. Now, this was a surprise to me, and most recently, we have now direct evidence from a high-grade glioma triggering spreading depression. Here, there is no direct brain injury, right? Now, this is a patient with high-grade glioma. This is post-surgical scan, an electrode is placed, and spreading depression is detected, as you can see here, okay? Now, this study was predicated on an animal study from Jeff Nobles in Texas, where they used a CRISPR-based mouse model of glioblastoma and did intracranial recordings, chronic recordings, and detected spreading depressions in this mouse model. So somehow high-grade glioma triggers spreading depression. We all know that they are prone to seizures. And what I think is that the seizures are culminating in spreading depression as the trigger events. So this is the definite. What about the probable? I'm going to do a quick sequence. What time? Okay, so I have 15 minutes. That's good. So this is going to be a quick flip the titles of the papers kind of thing, okay? We all know TIA can be a migraine aura mimic, and migraine aura can be a TIA mimic, right? We always have a hard time distinguishing the two, but there is a whole host of studies out there, usually single case reports or case series, reporting migraine aura-like transient events in patients who were discovered to have a vascular lesion to trigger them, and when the underlying lesion is treated either by surgical revascularization or by anti-thrombotic treatment, the spells stop completely. Okay, that's how we know there's a causative link between the ischemic source and the spreading depression trigger. Now, this is a particularly interesting case where the patient was a graphic designer, was having frequent visual migraine auras, typical visual migraine auras coming in several times a day. And this was a spell that he was having just before he got the MRI scan, and he wanted to actually draw it and tell people how it felt like. Okay, and this is typical migraine aura, this distortion of the image at the wavefront, and then there's a missing portion right in the middle. That's the scotoma portion here. Okay. This patient later was found to have a vertebral artery dissection, and upon institution of antithrombotic treatment, all spells stopped. Vascular malformations and the relationship with aura even has more publications out there. Okay. I'm not going to talk about every single one of them, just to give you an impression that this is a real relationship. Okay, vascular malformations can trigger spread depression. I have a case later, if I have time, that I'll show you. Let's not forget cerebral amyloid angiopathy. I was really fortunate enough to be part of this writing group led by Eric Smith and Steve Greenberg a few years back, where we argued that the transient focal neurological episodes or amyloid spells are in fact, at least some of them, based on the semiology, the way that they present, could be spreading depressions. And last but not the least, other forms of brain tumors have been associated with frequent migraine auras or spreading depressions. And they range from astrocytomas to meningiomas to metastatic brain tumors. There's one particular case that sort of caught my attention. This is a 33-year-old female, new onset episodic visual aura for about 6 months. The aura description was exactly what you would expect from a visual migraine aura. The workup later showed a right occipital astrocytoma in this patient, and after resection of the lesion, the spells stopped. So I threw at you a whole list of clinical conditions where ST is likely to occur. Some of them are definite, some of them are probable. The list is likely to grow over time. Now, what I would like to do here is just to review quickly what we think of ST semiology might be. In the absence of invasive electrocorticogram recordings, we have to rely on ST semiology to actually recognize one in our patients. So this requires the knowledge of functional neuroanatomy because it propagates by contiguity. It doesn't jump from one part of the brain to a remote part by axonal connections. It just has to go by contiguity. So the symptoms have to make sense in terms of the sequence of their recruitment into the deficit. And we have to have an awareness of neurological diseases in which STs can develop, which is what I tried to do earlier. Now, the signature features of ST is a slow spread of signs and symptoms congruent with the known topology of cerebral gray matter. Okay, so that's the signature. Now, typically we see negative symptoms like the scotoma, but in cases with visual changes or with sensory changes, there may be a brief period of positive symptoms like the scintillation in front of the scotoma or pins and needles sensation right before the numbness sinks in. Okay, as it propagates at the wavefront. So this is another clue that you can look for. And as I said, it has to be have complete resolution of the symptoms. Okay, and that's important. Otherwise, you have a fixed deficit and you have to look for other things. Now, the points to remember are the more eloquent the brain region, the more conspicuous the signs and symptoms of ST, right? If you have an ST in the prefrontal cortex, good luck recognizing that, right? You may have a mood change. You may have, I don't feel like doing anything today. I'm a little confused. I have a brain fog. Right? This is the common term that patients use, I have brain fog. So it's not easy to tell whether this is a spread impression or not. The second point is the headache is not obligatory. You don't have to have headache. You can have auras without headache, for example, which I have. And remember that this semiology holds true only in tissue with normal function. If you have ischemic penumbra, where activity has already stopped because of partial ischemia, if there's a spread impression moving in there, you're not going to get any symptoms because the tissue is dysfunctional in the first place, right? So you have to have normal tissue that it propagates into. Now, based on all this, I and a number of my colleagues are now gearing up to start a push towards a new terminology. Just as we have primary seizure disorders or symptomatic seizures, we would like to propose, and this is not an absolute thing, We have primary ST disorders and symptomatic ST disorders. Primary ST disorder would be a migraine aura, typically, right? And symptomatic ST disorder is when there is an underlying structural pathology triggering the spread depression. So you could perhaps separate the two. It would be important to separate the two because the second one has implications clinically, right? You don't want to miss those things. And to distinguish, primary ST disorder are usually occipital or visual in case of migraine aura, for example. And symptomatic ST disorder can be anywhere in the brain depending on where the pathology is, okay? Primary ST disorder often changes side. Migraine aura comes on the right or left from one attack to the next, but symptomatic ST disorder has to be side locked. It's always on the same side, highly stereotypic. The frequency of primary ST disorder is relatively low. If you have migraine auras, it doesn't come 3 times a day or 3 times a week. Usually it's much less frequent, once a month, once every 2 months, and so or so. But symptomatic ST disorders can be really highly frequent and come in clusters. Primary ST disorders often start younger. Of course, as you can imagine, symptomatic ST disorder with underlying structural pathology can start in older patients as well. And primary ST disorder or migraine aura has common triggers that the patient can identify: physical exertion, visual stimuli, and so forth. And there are some predisposing factors, just like in migraine aura: sleep deprivation, fasting, stress, and so forth. And the symptomatic ST disorder, the critical point here is that it has to somehow resolve after the treatment of the pathology. If you start antithrombotic treatment, you should stop having auras, for example, if it's working. Now, I have 3 cases, but I will skip to the last one in the interest of time. This is a 21-year-old, previously healthy female, referred to a tertiary headache center due to frequent migraine attacks, 3 to 4 per week, highly frequent migraine attacks. This has been happening since age 11, but has gained frequency increased lately. That's why she came to attention. She describes her visual symptoms as typical for visual migraine aura, spreading from center to the periphery over 20 minutes and so forth. But in 2/3 of the spells, the symptoms progressed to involve also sensory disturbances on the right side of the tongue and the right side of the face and right hand over 30 minutes. And occasionally she had speech difficulties as well, in addition to those symptoms. Symptoms were always on the right side. Patient was diagnosed with migraine aura. Migraine with aura. And a subsequent research MRI revealed a left parieto-occipital AVM, which was then treated, and the symptoms and spells that she was having since age 11 completely stopped. So clearly, migraine with aura and the other cases, if I had time, would underscore the same thing. They were all diagnosed with migraine aura. This was a chronic subdural after evacuation. And this is a carotid occlusion giving watershed ischemic lesions. They all were labeled with migraine with aura. So I will finish with this last slide by saying that migraine with aura is a misnomer to describe symptomatic ST. If you have an AVM and developing frequent visual auras or sensory auras, whatever that may be, and you call it migraine aura, that has nothing to do with migraine. Migraine is a primary headache disorder. There should be no underlying lesion in migraine. So you're misdiagnosing the patient. And it's dangerous too. You may miss a pathology, underlying pathology, if you cannot distinguish symptomatic ST from a primary ST. The problem is, and that is the elephant in the room, we call it migraine aura because of lack of anything better to call it. But why not use the term spreading depression more liberally? This could be spreading depression. It should be in EPIC as a potential, and dizziness is in EPIC as a diagnosis. You can pick dizziness as your reason for seeing the patient. Why not spreading depression? You know, it's a more scientific term, right? So as with seizure, we must acknowledge spreading depression as a clinical phenomenon. And we must teach medical students, residents, fellows what spreading depression is. It shouldn't be a grand round given 20 years after I start attending this on the service. You know, we should just put the medical chapters in medical textbooks. It should be in medical school curriculum. So we are trying to achieve that at the end. Hopefully 80 years after Leo described it. It's not an easy sell, as I said, but it's worthy. Thank you. All right. Yes. So yes, I'll start from the back. Sorry, guys. Sorry. Yeah.
People describe recurrence.
You said recurrent?
Recurrence. Yes. Recurrence spreading results.
Yes.
One after one. Is it possible that each recurrence spreading results relate to the creation of force? Like something different, what changes that it was in the level down, then like Noxia, and then
Yes. Yes. I'm sure you're familiar with the work by Stephen Schick. Oh, I'm sorry. Yes. Okay. Sorry. So do you know Stephen Schick? His work. This is exactly what he was modeling. And if you look up his papers, the balance between tissue hypoxia and he tried to introduce blood flow and ion changes and extracellular space changes and so forth. And what I have to tell you is that spreading depression comes in clusters only in case of severe brain injury, in subarachnoid hemorrhage, in focal cerebral ischemia, for example, in ischemic stroke. It comes in clusters, sometimes TBI, right? Outside of that context, it's usually single, not recurrent. Migraine aura, 2 visual auras back to back is extremely rare. I'm not saying it never happens. It's extremely rare. And if it ever happens, I might challenge it. Challenge meaning that, that I would look for an underlying lesion and call that a symptomatic ST disorder as opposed to primary ST disorder. Yes.
So, Cenk, is there any consensus about what the nature of the increased conductance is in the pathophysiology?
Yeah.
I don't know.
That's the holy grail at the moment in ST research. So people have done, I'm sorry, so yeah, so I have to remember to, so the question was, is there any research on what this elusive conductance, large conductance that starts the spread depression is, what mediates it? And the short answer is no. We don't know what it is. There has been a lot of research and we used not we personally, but everybody else in the field used every known channel blocker to date. And none of that prevents spreading depression. Okay, tetrodotoxin, any voltage gated calcium channel blocker, large conductance, low conductance, whatever it is, no matter what people try, even ions, that cesium, barium, will not prevent spread depression from happening. Once it's triggered, it just goes like nothing happens. Now, there's one theory, and it's only a theory, that the sodium-potassium pump transforms itself under certain circumstances into a large conductance non-selective channel. I don't believe that the data is enough to call that, but David Andrews in Canada is a big proponent of this idea. So we'll see if he comes up with any data to convince us, but we don't know the answer. Yes.
It's great talk.
Thank you.
This is a little bit of a crazy question, but we talked briefly about occasional positive symptoms that you had just at the beginning before it goes negative. The setting of brain tumor, for example, do you think was going to be the genesis peritumoral seizure? That positive and good goes out of control and realms, whatever it's running.
Yeah. So it's seizure and ST interaction is very complex. I have to admit that first of all, and I don't quite understand it myself fully. But even when there's no seizure, when spread depression propagates, at the onset of the depolarization wave, right, the DC shift that I showed you, at the onset of that wave, there's a small amount of burst activity. And we believe that is the burst activity that gives the positive symptoms, because cells are getting depolarized, but they are not under depolarization block yet. And there may be a potassium wave heading forward, right? Just initiating the depolarization elsewhere. And that is giving the positive symptoms. When we say positive symptoms, it could be paresthesias like pins and needles, or visual scintillations. Now, it's quite possible that peritumoral seizure activity is always there and giving the positive symptoms. And every now and then, it then triggers a sprain depression because seizures definitely trigger sprain depression. And it may be actually a protective mechanism, right? You would rather have a migraine aura than a generalized tonic-clonic seizure, I assume, right? So everybody would probably agree with that. So, so it's possible that there's an interaction of seizures and sprain depression in that setting. But the typical positive symptoms that we are talking about is usually in the absence of any seizure activity and linked to that initial depolarization, mild depolarization stage that triggers a burst of firing in some neurons. Yeah.
So thanks again for the amazing talk. So at the beginning, you kind of showed this diagram about how, we develop how the polarizations develop. And then we kind of have the ATP system and the exercise, which we kind of clear things out.
Yes.
And help, the transit swelling that can sometimes occur. So in cases where ATP is damaged, like ischemic stroke, where you don't totally have that mechanism intact, and these depolarizations that come after ischemic stroke be a source of secondary injury.
Yeah. Okay. Well, I mean, again, that's a great question, and that's what we thought for a very long while. That's what we thought. That's why sprain depression field gained so much momentum, because we thought that that would be the source of secondary injury progression in both ischemic stroke and subarachnoid hemorrhage, because there you have partial ischemia as well, right? Now the data is mixed on that. In concept, absolutely. I mean, it's absolutely possible, right? If you do sprain depression and make an animal hypoxic, the sprain depression may never recover, and eventually you may get cell death, right? So the two together may be sufficient to cause cell injury. But when you actually do the key experiments in vivo, when you look at focal ischemia, for example, and induce a lot of sprain depressions from outside, you don't get the infarct bigger. So that way is definitely entering the penumbra. Penumbra is definitely partially ischemic, right? But it just doesn't grow the infarct. So there's a lot of controversy. I'm on one side, a lot of other people are on the other side. So I'm going to leave you right in the middle. It could be, but we don't have incontrovertible proof for that. Now, let me say this. If you look at the changes, ionic changes and cell volume changes and whatnot, by themselves, they should not necessarily kill the cell, right? There is a lot of ATP consumption, oxygen glucose consumption, but you could, in theory, stay in that state for a pretty long time, right? I mean, you can bathe in high levels of potassium, you're depolarized, you're not doing anything, you're just sitting there because you're quiet, depolarization block. But maybe it is a protective state because mitochondria shut down as well, right? Maybe you want to just freeze yourself like that under situations of duress. So, so it's all bets are off. Yeah, and why don't we talk after? Why don't we talk after? Yeah, just you come down. Okay, I don't want to keep everybody. These are from my lab. One of them at least. So that's fine. We can talk offline. Thank you so much. Yeah, thank you.